of the National 5 Biology course. The topic is called DNA and the production of proteins, and on the board is all the information that you will need to answer the questions on this topic. Firstly, the first thing I want to talk about is the shape of DNA. You are very often asked in exams what shape DNA is. Double-stranded helix is always going to be your answer. What that means is it's two strands of DNA twisted around one another and connected together by what we call complementary base pairs. The four bases which make up the genetic code of DNA are adenine, thymine, cytosine and guanine. You do have to name them, you will have to know the names, but they're usually represented by letters in the questions. Wherever there's an adenine on the complementary strand there'll be a thymine, and whenever there's a cytosine on the complementary strand there'll be a guanine. Very often a question will ask you to calculate a number of bases based on the number of the other base. So for example, if you're told how many thymines there are in a chain, you know that the other chain has the same number of adenines. That's how complementary base pairing works. Right, in terms of what DNA actually is, where it's found, and a couple of words which people generally do find difficult. DNA is found in the nucleus of the cell. There are structures that are made of DNA are called chromosomes. A human cell, for example, has 46 chromosomes. We inherit 23 of them from our mum, 23 of them from our dad. And on those chromosomes are little sections, and each of those little sections is called a gene. And the gene contains the code for making one protein. That's what DNA is. It contains the information for making proteins. Each gene codes for one protein. You'll also notice in this cell that I've drawn, I've labelled the nucleus and I've also labelled ribosomes, which will be relevant in just a few minutes. So bear in mind, when we're talking about a gene, it's a section of a chromosome. I've attempted to see how these ideas connect together as well. The gene consists of A's, T's, C's and G's, that's the DNA bases. The bases then tell the ribosome what amino acids to stick together to make a protein, and the protein, the knock-on effect is that gives us a characteristic. So for example, having blue eyes, having curly hair, being left-handed, those things are all associated with the proteins we are producing. Now it's important that we understand how these ideas link together. What we need to understand is that the sequence of DNA bases, that's the order of bases A, T, C, G, etc. That sequence of DNA bases decides the sequence of amino acids in the protein that's being made. So if there's a different sequence, you're going to make a different order of amino acids. The different sequence of bases leads to a different sequence of amino acids, which leads to a different protein. That's why when you think back, or when you think forward to when we learn about Unit 3, we learn that mutations, which are changes to the DNA sequence, they will actually change the protein that's made. Now what you'll notice is this statement here, that mRNA carries the genetic code. That's one last thing that you need to understand. Said I've mentioned these ribosomes again. This is where the information to make protein is found in the nucleus, but that's where the amino acids are joined together to actually make the protein in the ribosome. So somehow the cell has to get that information from there to there, and it does that using mRNA. The M stands for messenger. You don't need to know that, but you do need to know that mRNA carries the genetic code from the nucleus to the ribosome where the proteins are made. And that is everything you really need to know for the third key area of Unit 1.